Hello, this is part 1 of the recording for chapter 5 on infectious disease. I'm going to share my screen, and if you could call up your PowerPoint. We'll start here. Key concepts. The first part of this lecture, we're going to go over things in general about infectious agents. And then the second half will go into the details of specific infectious agents and go into a little bit more about each one. So in general, we've got microorganisms all over us. Primarily, they do not cause infection. For the most part, the microorganisms living on us do not cause an infection. They colonize us. They, in fact, help protect us from some infections. However, they do still have an impact, and especially in low resource countries, developing nations, infectious diseases still kill off a lot of people. In this country, things like heart disease are more likely to take our patients, but around the world, infectious diseases still ranks very, very high in causes of death. I'm not going to read every word on this because you guys know how to read. All right, in general, let's go over the different categories of microorganisms. We have things, this is a terrible term, non-living particles. It means it's a thing that doesn't follow the rules of other living things. So for our purposes, we're going to focus on viruses. In this category, you could also include prions, which are the things that cause mad cow disease and the neuropathies that are associated with that. The prokaryotes, remember the difference between prokaryotes and eukaryotes. We are eukaryotic organisms. Each of our cells has its own nucleus. The prokaryotes include these tiny cellular organisms. So they're more complex than the viruses. They're still small, and they do not have a nucleus, but they do have genetic information. So then some single-celled eukaryotes, yeast, for example. Yeast are also classified as fungi, but yeast are one cell big, or can be one cell big. And then there are some protozoa that are one cell big. The one that we're going to talk about is the one that causes malaria. And then there are multicellular organisms that I'm not going to talk about at all, for example, worms. Now, how we identify pathogens. We take we take a sample from tissues and then culture that sample, try to grow up that organism, and use microscopic identification for some things like, for example, the protozoa that cause malaria. There may be a blood smear. You have to look through the microscope. It's not something that we do very often around here. These two are more common. So assays of pathogen targeting antibodies. What that means is that there are some rapid tests. They develop pretty quickly. We can have the results pretty quickly in the clinic. And what they're looking for is actually the development of antibodies that the patient has developed, some defenses that we've started to develop. This is often the difference between those rapid tests and the things that you send off to the lab. What's happening with the stuff that gets sent off to the lab is nucleic acid amplification. So we're looking for the DNA of bacteria, or if we're talking about a virus identification, depending on what the virus is, viruses have the ability. Some viruses have DNA as their genetic information like we do, but other viruses have RNA as their genetic information. And in either case, that nucleic acid can be copied and copied and copied by something called polymerase chain reaction. You may have heard of this, PCR. And that's the kind of amplification that we're talking about. So we're basically looking for essentially a fingerprint, if you will, of these species, things that we know to look for. We just have to get more copies of that genetic information. And that's where the amplification comes in. And that's not something that we can typically do at the bedside. Let's see, rising numbers of drug-resistant pathogens. This is something that you've all heard about it, antibiotic-resistant bacteria coming up. There's not an enormous amount that can be done about it other than being careful about when we prescribed antibiotics, more generally antimicrobials. But we'll talk about why it happens. These are the species that I would like you to know from this chapter. There's a whole lot of bacterial species, and I promise we're going to go through each one individually. There are some viruses here in the center, and finally the one protozoal microbe that we'll talk about is the one that causes malaria. This slide is just making the point that in low-income countries or low-resource countries, infectious disease is highlighted here. These are the top 10 causes of death in the different areas of the world, and notice how many of them are related to infectious disease. In our country and in many high-income countries, lower respiratory infections still take a lot of us, but it's really mostly other diseases that take the life of our patient. This diagram is quite simple, but I like the point that it's trying to make, which is this. At birth, we are exposed to microbes. The vast majority of them are colonizing microbes that are in a commensalism. That is, the organism benefits from being part of us, but we are not damaged in any way. In fact, in some cases, the microorganisms provide a service for us. When there are microbes that are not beneficial and are having a harmful impact, we call that an infection. Mostly those are other organisms. In some cases, these precipitating factors, in some cases, some of the colonizing organisms can create an infection. For example, we have E. coli living in our guts, and that's a great place for them to be. And they do stuff for us and help us out. However, if they migrate to the vagina or to the urethra, they can cause an infection. So that's what I'm precipitating factors. Other things can happen. We have, for example, yeast and bacteria both growing in the vagina, and they balance each other out. If somebody ends up taking an antibiotic, in many cases, there are many women who end up suffering from a yeast infection after that because the balance between those organisms has been tipped by a medication. And again, that's what we mean by precipitating factors. Now let's take a look over here at infections. Some infections get cleared by our immune system. We call that resolution. Sometimes we have a chronic infection that we cannot get rid of even with treatment. Examples of that include HIV currently, herpes, for example. Mostly it's viruses that we're not really great at getting rid of completely, but there are some chronic infections, like for example, tuberculosis, that people will live with for years without it progressing necessarily to a disease state. If the infection cannot be controlled, the infection can lead to death. Okay, moving on. This chart is basically saying the difference between colonization versus infection. You understand the principle. If it doesn't hurt you, then it's colonization. If it starts giving you evidence of inflammation or other symptoms, that's an infection. What impacts whether something stays as a commensalism or a colonization versus progressing to an infection? A lot of that has to do with the level of health of the patient and how robust the immune system response is. As we'll see next week, in the very young patients and in our very oldest patients, these are often the most vulnerable to infection because the immune system is not as strong as it is at other times of life. If the patient is undergoing immunosuppression, either because of an infection like HIV that actually brings down the immune system, or if they have to be on medications that disrupt the immune system. For example, they're undergoing chemotherapy, or they have to take steroids for some other reason. Both of those are going to suppress the immune system, and the patient's more likely to develop an infection. The other case, and this one's the most common around here, is having poorly controlled diabetes. That is a chronic disease. It has known effects on suppressing the immune system. And so it impairs the immune defense against infections. And so patients with uncontrolled diabetes are frequently more at risk of developing infections. As we did last time, each time you see this buff colored background, we are switching topics. So I'm going to talk very generally about characteristics of bacteria, and then we'll talk generally about characteristics of viruses, etc. So briefly, these organisms are small. They're less than 1/10 the size of a human cell, and some estimates less than 100th of the size of a human cell. They reproduce really fast. Sometimes you can get 3 generations in an hour. So we're talking about exponential growth. You know, all the bacteria just seem to arrive at a good place, and then they just start making copies like crazy. It's a very fast process called binary fission. It's not as complex as mitosis. All right, so talking about structure, we have cell membranes, so do bacteria. However, bacteria, in addition to the cell membrane, they also have what's known as a cell wall that we do not have, and that's outside of the cell membrane. You'll hear the bacteria referred to as gram-positive or gram-negative. We now know that that's related to their classification. When the technique was first developed, it just had to do with like, what color does this thing turn? And when you look at it under the microscope, does it look pink or does it look purple? But you'll still hear this term, gram-positive or gram-negative. Characteristic shapes, there are some terms that you're going to encounter again and again. The coccus refers to a ball-shaped structure. And then the prefix, strepto, means that it appears in long chains, like a strip. Staphylo means it appears in a cluster like grapes. So you'll hear staphylococcus or streptococcus. You'll hear streptobacillus. Bacillus is this rod-shaped. And the one other thing you might hear is spirochete. It just, the bacterium itself is kind of corkscrew looking. Little anatomy for bacteria. Again, let me remind you, there's a plasma membrane. This is also called a cell membrane, phospholipid bilayer. Then outside of that, there's the structure that we do not have, and that's the cell wall. And this is the target for many antibiotic medications that go after bacteria. And it's a great target because it affects the bacterial growth, but it doesn't affect our cells. In case you heard that, that was my dryer. And then outside of that, there is a capsule that some species have that make them even harder to get at. We mentioned that they duplicate by binary fission, so the doubling rate, the generation time, it's fast. And so you get a huge increase in cell numbers very soon after infection. Then here's an important point to understand. There are so many infections that seem to all have the same symptoms, and it seems like, why those symptoms? Well, it turns out those symptoms are actually not directly related to the organism that's causing the infection. It's related to our immune system response. And that's why they so frequently seem like exactly the same symptoms. So the fever, the increasing body temperature, malaise, the general feeling of being run down, anorexia, you lose your appetite. That's again related to our immune response. That's why all those flu-like symptoms are so common for all of these different infections. I'll let you read more about that. All right, so here's an important principle to understand. There are antibiotic drugs. This term, I know if you break down the term itself, it literally means against life or against living things. However, the term use has morphed over time. So when we say in a clinical setting, we say antibiotic, it means very specifically antibacterial medication. So again, don't go off of like the Latin says that's just against life. Just think this word now means against bacteria. It does not mean against viruses. It does not mean against fungi. If you're looking for a term that means against all those microorganisms, you'll hear the word antimicrobial. That's the umbrella term that includes antibiotics in it. All right, so let me just mention a few targets. This cartoon is of a bacterium. Some of the major targets for antibiotics are the cell wall. I mentioned that we don't have a cell wall, so that makes a great target. Examples of medications that do this are penicillin, one of the first antibiotic, but now the more modern version of that is amoxicillin. But they're both what's called beta-lactams. And what those beta-lactam rings do is they basically pretend like they're building blocks. And so when the bacterium is trying to make copies of itself, it puts its building blocks, they're called peptidoglycans if you care, and then it takes a beta-lactam and it puts it in there, and later that beta-lactam is not a good building block. And so that cell wall kind of falls apart and is open, and that bacterium cannot survive. Yay for antibiotics. As we'll see, that doesn't always work, and I'll explain why later. Other targets, nucleic acid synthesis. Yeah, we have to copy our DNA and RNA, but the bacteria do it in a slightly different way. And so that's a target for antibiotics. For example, Cipro, if you've ever administered that, you're messing with the bacteria's ability to make DNA. And then finally, protein synthesis is another good target because our ribosomes are just slightly different from some bacterial ribosomes. And so these make a good target. And an example of a medication that targets that is clindamycin, in case you've ever administered that one. Okay, back to the buff colored. So we're moving on from bacteria and antibiotics. Now we're moving on to viruses. These are the super tiny organisms that can cause infections. Remember we said bacteria were less than 1/10 the size. These are even smaller than that. Less than 1/10 the size of a bacterium. I mean, they're minuscule. And they're mean and lean, and I really am so interested in these. They have nucleic acid, so either DNA or RNA, depending on the species. And they're nothing but business. It's like, that's it. That's the DNA or the RNA. And then we've got this protein coat around that. And for some bacteria, for some viruses, that's it. That's all you have. Other viruses, you may see something that looks familiar. Many have this membrane-like envelope, and they actually steal it from us. It's a phospholipid bilayer, and they take it from our cells as they bud off from our cells. But this might look familiar. You've heard talk of the spike protein of COVID-19, of the SARS-CoV-2 virus. That's where the spikes are, is on the outside of this envelope. Okay, depending on the species, the protein coat is a different shape. Typical viral life cycle. Viruses have nothing. They do not have ribosomes. They do not have mitochondria. They don't have any of that stuff. They travel light. That's how they can get away with being so small. And this is in very general terms. A virus that's just greatly exaggerated in size here arrives at a cell, and it has to get its nucleic or its DNA or RNA, nucleic acid, into the cell. Sometimes the whole virus comes in, sometimes it just injects the nucleic acid in. The RNA viruses can go from there to just start transcribing and then translating. When I say they transcribe and translate, they don't do that. They just say, here's some information, do this for me. And we do. Our ribosomes will do the translating of that. We make their proteins for them. Genius strategies. All right, at any rate, whether it's DNA or RNA, the end result is that you're going to make some proteins for them, and then they get us to package that stuff up for them. And so that's, remember I said the envelope comes from us. Yeah, this is that budding off and making more. So then those viruses go off and infect more of our cells. All right, key elements I've mentioned have DNA or RNA. They have to attach to the host cell. All right, here's the thing. I've said this repeatedly. Viruses carry almost nothing. And so there are precious few targets on viruses for going after. They use our cells kind of like human shields. Like they use our ribosomes, so we can't target their ribosomes because they don't have any. And so the best defense against viruses is vaccines. Vaccines allow our immune systems to see some bit of the virus so that when we are exposed to the virus for real, we've already had a chance to kind of ramp up production of B cells and T cells that are going to go after that virus. How the mRNA vaccine that is being used currently against SARS-CoV-2, how that's different from the regular vaccine, like how the Pfizer vaccine is different from the Johnson & Johnson vaccine. The mRNA vaccines, remember back here when I said the virus introduces its RNA and then we transcribe and then translate that RNA and make the proteins? That is what the mRNA vaccination does. It's nothing that viruses haven't been doing for millions of years. It's just that when we introduce it into somebody's arm, we're giving them just the instructions for making just the spike protein. We're not going to make the protein coat. We're not making anything else. We're just making that one spike protein, because that one spike protein is the key for the virus finding its way into our cells. And so we say, if anything's going to change, that's the last thing that virus is going to change. So let's focus on that one. And so that's what we do with that RNA vaccine, is just making that one protein so that our immune cells can learn that one protein and can go after it when it's time. All right, and that's our best defense against viruses at this point. There are some antiviral medications, but there just aren't nearly as many antivirals as there are antibiotics because there aren't as many targets. So we don't have as many effective treatments against viruses. Our best treatment is actually a defense, which is here, introduce a little bit of the virus to the patient so that when the patient actually encounters the virus, the immune system's already been primed. All right, now we're shifting gears again. Key elements of fungi. So far we've gone over bacteria and viruses. We're moving on into the fungi. These are eukaryotic cells. So they're like our cells, they're big like ours, and they have a nucleus, and a lot of the structures are the same. So again, we're in that same problem of how are we going to fight against this, because these cells are so similar to our cells. Well, one way is, you remember that our cells in the phospholipid bilayer, we have the phospholipids, we have proteins, and then we have cholesterol. Well, fungi also have phospholipids and proteins, but their sterol, instead of using cholesterol, their sterol is something called ergosterol. It's a slightly different shape, and it makes a fantastic target for antifungal medications. And so largely that is how we deal with fungal infections, is by going after the sterol that's different from ours. Here's another important point. Fungal infections, a lot of them are superficial. They're on the skin. If the fungal infection becomes systemic, that's usually a bad sign. Mostly patients' immune systems are able to fight off fungal infections. So a systemic fungal infection is less common and more lethal. It's more serious. Often the systemic fungal or systemic yeast infections are more common in immunocompromised patients. Because I only have one slide for fungi, I put this one in to really make it clear. We're shifting gears again, and now we're moving to protozoa. We're nearing the end of part 1. Protozoa, like fungi and like our cells, are also eukaryotic. There are so many different species of protozoa, and the vast majority of them are not pathogenic. They do not cause disease in humans, except for this handful that's listed right here. The ones that I'm going to talk about in this lecture are malaria. I am going to mention in passing giardiasis, because for those of you who are here in the desert Southwest, this is something that we do see. Giardia grows in natural water sources. And so you'll see this in people who have been camping and hiking and drinking the water without filtering the water, without boiling the water first. And it leads to gastrointestinal upset and diarrhea. But for our purposes, for this lecture, we're mostly going to focus on malaria, and very briefly at the very end of part 2. And there, clinical topics. All right, so we are identifying these organisms. So direct identification, microscopic examination. The term culture means to grow it up in the lab. And then these two things we've already mentioned, the antigen identification, those are usually the rapid tests that we can do. What it's testing for is the presence of antibodies. So our own immune system kicking on to start fighting off that infection. Those tests are not as accurate necessarily as the nucleic acid amplification testing, the NAAT. In that grouping, you'll also include the PCR test, the polymerase chain reactions that help to amplify the DNA and RNA that you're looking for. And those are the tests that take a little bit longer to get turned around. Antimicrobial resistance. So a lot of our pathogens have very fast generation times. They have high mutation rates. Those are perfect conditions for favoring the development of drug resistance. I mean, these organisms are not doing anything evil. They're just doing what organisms do. You know this, natural selection. There's a variation. The environment changes. Some variations are going to get selected against. They're going to get killed off. And other selection or other variations don't get killed off. Those are what we say are being selected for. Before I get onto this point, it is not a huge surprise that anytime a new antibiotic comes on the market, after some time it's being used, you're killing off the organisms that are susceptible to it. If there is variation in the population, if mutations arise that allow some of those organisms to survive that antibiotic, amazing. That is being selected for. I know it's not happening on purpose. We're killing off the things that are susceptible, vulnerable to our antibiotics, and we're leaving behind the ones that aren't killed off. And so those are the ones that survive and reproduce and go on to have, a small colony of antibiotic resistance. And then they pass that along to their offspring. What makes it even harder is this trick that bacteria can do that other cells cannot do. Bacteria have this ability to not only pass new genetic mutations on to the next generation, they also have what's known as horizontal gene transfer. They have these little rings of DNA, small bits of DNA separate from their main chromosome. They're called plasmids, and they're able to pass those to each other. So if by chance one of them has a mutation that makes them more resistant to a particular antibiotic, it's not just that particular bacterium you have to worry about. That one is passing on information. It may be also passing on terrible information, not helpful information to other bacteria, but that drug resistance can pass within one generation, can pass to other ones. And so that's how you have drug resistance show up and spread like wildfire in a population. It's really quite interesting. So antibiotic stewardship means be careful. Don't prescribe antibiotics when you don't need to prescribe antibiotics. If the person has a viral infection, you suspect that they have a viral infection, don't give them antibiotics. They don't need it. And you're preserving the integrity of that antibiotic, if that makes any sense. Mechanisms of antibiotic resistance. All right, this is why I went into that detail about the beta-lactams, like penicillin, amoxicillin. They act like the little building blocks of the cell wall. All right, great. Some bacteria have developed an enzyme that clips that little beta-lactam ring. So here we had this great trick that we said, here, build your cell wall with this. And now we have bacteria that can snip that. It's called a beta-lactamase. ACE means it's an enzyme that does something to beta-lactam. So we have the antibiotic amoxicillin. Now I've got this antibiotic resistant strain of, let's say, bacteria growing in the kid's middle ear. Okay, we know that trick. So then, just like evolution happens, we humans have come up with a comeback to their beta-lactamase. We have a medication called a beta-lactamase inhibitor, which crushes the little snipping tool. So we say, here, I'm going to give you a building block, and that tool that you have that's going to destroy my building block, I'm going to crush that one. And so if you've ever administered to a patient amoxicillin with augmentin, that is what you're doing. You're saying, I'm going to give you this building block, and I'm going to give you this thing that in case the bacteria that's growing in your ear has that snipping tool, we're going to crush that so that we can still use this antibiotic. And so it's going to go with development of antibiotics and the evolution of antibiotic resistance. This is going to be an unending battle. And then bacteria have other techniques that they can use to defend themselves against these antibiotics. And these are the species that we are going to cover in detail in part 2 of the lecture. And we'll start here for part 2. So I'll end my share.